Hi everyone, welcome back to Eclipse Biology Discuss. In my previous lecture of cytology, we have discussed about the detailed structure of prokaryotic cell, and in some lectures we have discussed about what is the constitution of the cell wall in case of prokaryotic cell. In case of prokaryotic cell, what is the constitution, what is the structure of plasma membrane, as well as we have discussed about the capsule. Then we have already discussed about the mesosome and ribosome. Okay, and in this lecture we are going to discuss about the nucleoid or genophore. What is nucleoid? Actually, what happened in case of your prokaryotic cell, there is absence of a true nucleus. We know that. So that we call the cell as the prokaryotic cell, means primitive, or there is presence of a false nucleus. Karyon means nucleus, pro means primitive. Okay, in case of prokaryotic cell or a cell that is having a false nucleus, that is called as prokaryotic cell. Thus, here in case of prokaryotic cell, there is absence of a true nucleus. But as we know that in case of your nucleus, there is presence of what? There is presence of genetic material. What is that genetic material? Genetic material means that is the DNA. Okay. So in case of your prokaryotic cell, there is absence of nucleus, but the main constituents of the nucleus, that is the DNA, is present in case of prokaryotic cell. Suppose this is a cell. Suppose this is a prokaryotic cell. Okay, I am not concerning about the different parts of prokaryotic. Just let's see what is that nucleoid or genophore. Okay, in case of prokaryotic cell, suppose this is a cell. This is the cytoplasm. In the cytoplasm, there is presence of a double-stranded DNA. Okay, there is presence of double-stranded simple DNA. Okay, and where this DNA is present, that area is called as what? That area is called as nucleoid, or we may call this as genophore. Okay, so this area that is called as nucleoid or genophore, and this is what? This is the DNA. Okay, in case of prokaryotic cell, the DNA that are very simple and these are small as compared to eukaryotic DNA. Okay. In case of prokaryotic cell, there is presence of double-stranded DNA. There is presence of double-stranded DNA. In case of eukaryotic cell also, there is presence of double-stranded DNA. Okay. But in case of prokaryotic cell, there is absence of chromosome. Okay, here there is absence of chromosome. What is chromosome? Chromosome is nothing but this is made up of what? The chromosome that is made up of DNA. Okay, in case of eukaryotic cell, the DNA that is very large or very long, so that it requires its packaging. Okay, it undergoes packaging. Due to packaging, what happens? The DNA undergoes packaging and it comes to a packed condition. It comes to a packed condition. That packed condition leads to formation of a structure that is called as chromosome. But in case of your prokaryotic cell, there is absence of a complicated DNA. There is absence of a complicated DNA or very long DNA that is absent. Here there is presence of a simple DNA. Okay, and that is very small as compared to a eukaryotic DNA. Thus, it do not require its packaging. Okay, this is just present in the cytoplasm. Okay, and where this DNA of prokaryotic cell is present, that area is called as nucleoid area, or there is presence of an area that is called as genophore. That means in case of prokaryotic DNA, prokaryotic cell, there is absence of nuclear membrane Okay, there is absence of nucleoplasm and there is absence of nucleolus. Did you know that in case of eukaryotic cell, there is presence of a complete nucleus. Complete nucleus means there will be presence of a nuclear envelope, that is called as nuclear membrane. There will be presence of nucleoplasm, some viscous fluid, semi-fluid materials will be present inside the nucleus or in the nucleus, that is called as nucleoplasm. But here there is no separation of nucleoplasm and the cytoplasm. Okay, but in case of eukaryotic cell, the nuclear membrane it separate the cytoplasm and the nucleoplasm. Okay, but in case of your prokaryotic cell, there is absence of nucleoplasm as well as there is absence of the nucleolus. Okay, then in case of eukaryotic cell, there is requirement of a protein. There is requirement of a protein for its packaging. That protein is called as histone protein. That protein is called as histone protein. Okay, in case of eukaryotic cell, there is requirement of a histone protein for what? For the packaging of DNA. DNA undergoes the packaging and it will lead to formation of a compact structure that is called as chromosome. And that chromosome that is made up of DNA, and this DNA undergoes the process of packaging with the help of a protein that is called as histone protein. Okay, but in case of prokaryotic cell, there is absence of this histone protein. Okay, so another point is here, there is lack of histone protein in prokaryotic DNA. Okay, so this is all about the nucleoid or genophore. Okay, but sometimes you may have found in some bacteria there is presence of extra nuclear material or extra chromosomal material. Okay, this is the extra chromosomal material. What is the name of that extra chromosomal material? Yes, that is the plasmid, right? The extra chromosomal material, that is the plasmid. Okay, this is non-essential extra chromosomal circular DNA present in case of some bacteria. Okay, so this is the extra chromosomal material and this is a non-essential. This is non-essential extra chromosomal circular DNA. Okay, they have no essential role in case of your prokaryotic cell, but if it is present, it may possess, for example, it may possess some of the antibiotic resistance genes. And this is a DNA. This DNA will having the genes. Okay, it possesses some antibiotic resistance genes. If we are giving antibiotic against a particular bacteria, then it will be resist against that particular antibiotic due to the presence of some antibiotic resistance genes in the plasmid. Okay, so this is all about the prokaryotic nucleoid or nucleus person. Okay, you may not call this as nucleus. Okay, so that we are calling this as nucleoid. Okay, as there is essence of all these three components, so that we are not talking about the nucleus, so that here we are talking this as the nucleoid or genophore, or this is the false nucleus. Okay, this is the false nucleus. Okay, thank you. Hope you understand this clearly. Please like, share and subscribe my channel. Thank you.